Viewers, in this video, which is suitable for beginners, we are looking at the basics of transcription or transcriptional units. Transcription. So before going to what is transcription, you all know what is the central dogma of all biology, central dogma of life. So what does it say? It's DNA is made into multiple copies of it by the process of replication. Which the template DNA is decoded by the process of transcription into messenger RNA, which is then translation, the process of translation into the sequence of amino acid called proteins. So when you take, this is what you call the central dogma of all biology. So this process cannot be reversed. Protein cannot be used to make proteins, or proteins cannot be made to use the synthesis of mRNA. So this is the central dogma of biology. DNA. In the process of replication, so if we suppose it is the DNA, in the process of replication, the entire DNA is replicated. But what it is, in the process of transcription, a segment of DNA, take this, a segment of DNA is transcribed. That is, gene gets transcribed. So, take this, 3 dash, 5 dash, 5 dash, 3 dash. It has a promoter in the 3 dash region. This is promoter. And has a terminator at the end. And in between them is the structural Ds. So what is there in the 3 5 strand is called template strand. Whereas in the 5 3 region, it is called coding strand. So this is the basic structure of a transcriptional unit. Suppose in the coding template strand, if it is the 3 5 region, it is G, C, A, C, G. Then in the 5 3 region, which is the coding strand, it is C, G, T, G, and C. So this is what it will look like in the coding strand. So the coding strand is the same as the mRNA, but the thymine in the mRNA is replaced by uracil. Promoter region. What is the promoter region? Is the region where RNA polymerase binds. So this promoter region is anti-rich. So it's called starter box. So when it's an eukaryote, it is described as Hockness box in eukaryotes, whereas in the prokaryote is referred as Prignol box. So those two are named after the corresponding scientists who discovered them. Found in the upstream region, that is the 3' end of the template strand or the 5' end of the coding strand. So when you take terminator, it is where the process of transcription ends. So it is present in the downstream region, that is 5' of template strand and 3' of coding strand. So, and in between them are the structural genes. So these structural genes are referred as cistrons. So cistrons can be monocistronic or polycistronic. So when you take the monocistronic, which is a characteristic of eukaryotes, it calls for a single functional unit, whereas polycistronic, which is a characteristic feature of prokaryotes, which calls for multiple functional units. So many cistrons are found, and it calls for many functional units. A transcription process is associated with many functional genes, whereas in eukaryote it's polycistronic. A process of transcription yields a single functional unit. So more on this will be in the next video. Thank you.